Okay, good morning. So I actually did want to address this issue because Kevin texted me like in the middle of the night and he's like, I know that this isn't excused and I'm really sorry, and you know, like I'll take my punishment, my hanging, my lashing or whatever. And I realized that I didn't maybe do a good job. I think I presume too many things with you guys who've known me for so long that of course if you need to miss, you know, it's really not a problem. Just let me know. We're certainly not going to tie you to the stake or anything. Definitely just give me a text and it's no problem at all. So as I mentioned, I think there's a little bit of nerves that come, and I know for me, when I think I've got this writing project that I have to get done, and sometimes it's easy to feel like you're starting from scratch. And so I wanted to take this first part of our period today to just remind you that we've actually already talked a lot about the proposal. So at any given moment, feel free to go back to the syllabus, and do note that, you know, yes, it does give you so much of what we are doing in class. So when you get to the research proposal description, which is the first assignment description after the body of the syllabus schedule, you'll notice that what we've been doing over the last periods of our class has been to go through the sections of this. So we've already covered a majority of these sections. We haven't necessarily done it in order, but we've done it from what I consider the roots of the tree to the fruits of the tree. And so at this point, we've, so midway down the first page of the proposal assignment description, we've talked about the title. That was yesterday. Today, we will be covering the quote abstract, or in NSF, it's called a project summary, and it's actually a little bit different than an abstract. We began our coverage with the statement of problem significance or value, and that is something that all of you drafted, and you drafted it for both projects. So at this point, you can go ahead and think about making your significance statement for the project that you're on become a little bit stronger with your proposal submission. So this is not at all something that you're starting from scratch. It's something that you're working to build from your original version. Many of you also wrote a portion of the introduction. That is, not everyone, but a certain number of you definitely sent me a relevant literature section. And so this is something you've already begun. We haven't written the rest of the introduction yet, the preliminary data and empirical model, but that is something that I can see coming together even as you begin to work with your group. So it may be rough and drafty in your individual, but then when we work with a group, we might bring that together. And sometimes people are just very visually oriented. And so it very well might be that Claire decides to spend a lot of her time on her proposal draft on an empirical model, because you just see how this fits into the community. You know, you've got your Banner Road wastewater treatment plant, you know, human microbiome samples, poop samples, you know, how does that fit into characterizing, you know, to helping the downtown clinic where it's located over on 2nd Street, and you've got this flow diagram, kind of similar to the one that we looked at in the NSF proposal a couple of classes ago. And so if Claire, say, does spend a lot of time on that, then that would be the portion from her individual proposal that I would probably pull into the Google Doc and say, wow, she really did a great job of this. Let's put that in. But then maybe Josh, you know, and I already know, he spent a lot of time writing the relevant literature section. So his section there is already budding. It's already, you know, beginning to be a very strong section. So it might be that that is a section that I pull in for that group's relevant literature portion. So what I'm trying to say is that I actually don't want to put barriers on you. So say that you get to writing and you spend a ton of your time on one section versus very little on another. That's fine. I don't want to put barriers on you there, because I think that's one of the ways that we capitalize on where your greatest interests and strengths lie. But try to get something written down for each section, you know, that maybe will help us to build from. So turning to the next page, on the top of the next page, all of us have already drafted objectives, hypotheses, and specific aims. Now, we might not have been 100% happy with them, but we've already practiced them. So they're going to be things that you're building from and working off of. We already have talked about the research plan, and our goal for this week is to start to get an outline of your plan. That can be a visual schematic. So what I would like for you to do is, I'm gone on Thursday. I'll be traveling to New Mexico for the New Mexico Invitational. So when I'm gone, you guys are going to be working and bouncing off of John, and hopefully handing him even a handwritten research plan that's just an outline, or it's a schematic, or it's something that, and here, let me give you the objective here. John needs to know what to order. So we need to make sure that he has some idea of the extra reagents or media. You know, if we need to order a boatload of Jensen's agar to support all of the actinomycete isolations, or you guys are like, man, I found this cool agar that will allow us to isolate the acetobacter, could you order some? You know, that's the kind of thing John really needs to know. So we're hoping that on Thursday, by the time you leave class, and we're going to give you time to work on it in class, that you can hand him some kind of outline that gives him a rough idea of maybe the kinds of things he should order for you. So we've talked about that, right? We've talked about research plan. And on the back, timeline, something we're going to talk about today. So our goal today is the quote abstract or project summary and the timeline. And then we've already done references. So recalling on our tree analogy that we have already talked about each of those sections that we just went through. And I just want to review how they fit into the entire tree and our so-called growing proposal. So we talked to begin with about just getting started, having that big picture view, and having significance. We talked then about how to organize your writing and the 5 axioms of writing that are really important the whole while, right? Highlight, funnel, focus, unify, bringing together everything in a coherent and clear manner. We then went on and we talked about significance. We said that the root of the tree is your significance statement. That's why we did so much time on drafting those significance statements. So you guys already have what I would believe is a working draft of a significance statement. It might get better when you submit your drafty proposal, or it might be one of the sections you don't have time to work a whole lot more on. But I would say it's something you can think about. We talked about how we funnel, right, to our own research goals, so remembering those axioms of funneling. And then we talked about the introductory material, how the water for the tree is the references. You all have gotten at least 3 pivotal references, and today I saw Josh brought in another that might be pretty pivotal in typifying microbiome populations in poop. You know, it's definitely our pivotal source. And so we talked that that's the water, right? That's the water for the tree, and you already practiced writing those references up. In terms of your kind of nascent but beginning list of references. We then wrote objectives, hypotheses, and aims, talking about how your introduction funnels you to those statements, and spent a little more time on formatting of references. So this is all the water for that tree. We then mentioned that this is what allows that tree to grow. And the core or center of your proposal is the research plan. So, John, I was just mentioning to them that on Thursday they'll be trying to get an outline together that they'll hand to you with that intention that they will be doing, that you will be trying to do the ordering that at least gives us the basis of what is needed. Okay, excellent. So we talked about that research plan and the portions and sections of the plan. Although we're still in outline stages of that. So that brought us to the title, remembering that the title is more than you might think in really describing the project, and something that, as we talked so much about yesterday, can be pivotal in finding what you're looking for when you are searching for an article that's pertinent to you. So we talked a ton about that yesterday, and I won't go back into that, but I want to bring us to where we finally are now, and that is writing what NSF likes to call the project summary, or what NIH calls the abstract. NSF does like to distinguish its project summary from a typical abstract. And I'm going to read to you a couple of portions of the book that talk about the project summary. And I want you guys to think about, as I read through these, what are some of the major differences between what you hear here about the project summary versus what you would recognize as the typical abstract that maybe you're used to writing, because who's written, a couple abstracts before, right? Okay, cool. So I want to start with this one sentence because I love it. A convincing and exciting summary that captures readers' attention and interest, and it establishes a strong tone for the entire document. This is like what your reader would come back to get a flow for what your entire document is going to be about. It kind of summarizes everything in one fell swoop. So NSF's description of the project summary from the NSF sources, so this is a quote. It should not be an abstract of the proposal, but rather a self-contained description of the activity that would result if the proposal were funded. That is, if you're given the money, what the heck would you do with that money? Okay? It should include a statement of objectives, methods to be employed, the significance of the proposed activity, and insofar as possible, it should be understandable to a scientifically or technically literate lay reader. So anything in there that just steps out at you as being just a little bit different than an abstract?
Like exciting, engaged.
Yeah.
Abstracts are boring and droll.
Yeah, totally. The return to significance, the capture of the reader, right? All of these things sound a little different than maybe the mundane abstract that we're used to writing, but also good, right? It seems nice to think of writing something that might capture the reader's attention. So.
Aren't for the general public either.
Right.
Thick.
Yeah. Often an abstract is written very much to tickle the mind of other scholars. And this is something a little bit different than that. And I think intentionally so, because again, it wants you to return back to the broad implications of your work and to convince your reader that in fact it has interest for someone inside and outside of the field. So that being said, there's quite a lot that we can say about the project summary in terms of sections that you might include and then how you might write it, like format that you might use. So firstly, just to look at what it includes, it should have your goals, that is a statement of your objectives, your hypotheses and aims. Now, it might be some writers are so bold as to start it with, this proposal will test the hypothesis that. And that is one way that you can start it. However, that is not the typical funneling way that we've talked about. And I think in often cases, funneling can be more effective. So one or two sentences that captures the reader's attention and shows them that what you're doing is important. For the downtown clinic, we obviously know that we can make a very broad statement about potential implications of our work just based on our visit yesterday, right? So much that we can say that will capture the attention of the reader, but that then we do want to come in and talk about the objectives and hypotheses. So stating the context might be done very, very early, and it may be part of what captures your reader, but then stating the objectives and hypotheses will be important as well. There is no, quote, relevant literature portion of this, except for that we do want to establish the need for the research. So you want to be able to say that there is a gap in the knowledge. And you may even, at this point, and I'm going to jot this down, you may even find it appropriate to insert some sort of, I thought that I brought this up, to insert some sort of previous data, so preliminary research. Let me just pull this up. So I can write on here. Excellent. So we might just jot down, in addition to IDing the gaps in the knowledge, which may have a foundation in relevant literature, you also may want to put in something in this area that's like preliminary data, because you may be making the case that the work that you're capable of doing will fill in that gap. So again, we don't have a ton of or preliminary data or preliminary results. We don't have a ton of that, but we've talked about what it is that we might include. And I know we got a copy of the soil chemistry report to acres. That could be encompassed potentially, but so too could all that we did last semester be sort of a relevant preliminary data or results. And that would be very short within the context of the project summary. So once again, this is like, how do you do everything in one fell swoop? I mean, look at how much we're looking to include in this. It's like I put a star here. It's like you're putting the whole project in 2 paragraphs. And for NSF, they say, please don't exceed 1 page with this project summary. So it's really quite a short statement of absolutely everything that the project will encompass. So sentence-wise, right? Paragraph 1, stating the context, state the objectives, the hypotheses, ID the gaps in knowledge. This could go into a little bit of relevant literature in order to ID those gaps. It may also show how you might be able to fill them based on your preliminary results. And then succinctly describe the work that will be done. This is not a detailed methods section by any means. That's going to come later, right, in your methods section. But this is a statement of how you will do, what you will do, and how you will do the work that you will do. So the paragraph 2 will talk specifically about the techniques that you hope to use. And again, details will be omitted there. But your context, your study site, your organisms, and your projected results or outputs. This also is where this differs a lot perhaps from a typical abstract, is that in significance being a really pivotal or important part of this, we know that we want to draw the reader back to that broad import. And so remember reading that passage that was like, wow, you want to capture the attention of the reader. So that's an important part of that. Okay, cool. So I just want to write down in terms of significance, how the work will advance your area, or discipline, or whatever you might say there. The broader significance, and one of the things that often gets included in there, so like I might just say societal benefits. This is so easy for us, and I'm going to have you guys read some examples of other project summaries. And what you will find is that in many cases, the societal benefits, that is the teaching and training or community benefits, so I'm just going to say stuff like teaching, training, community. In many of the project summaries that we will look at, these things are going to be, you're going to be able to tell that they were forced. They were kind of forced into the plan in order to fit NSF. We are not doing that. Our entire project is based on the foundational significance and the community outreach. So we're in no way forcing the significance. We're beginning from the significance and we're building on the roots, right? The roots of the tree are our significance. And so that's a cool thing about your project, that you'll see some of them have to force a significance there. So teaching, training, community outreach, right? All of those things that might fit in there. Okay, so there's 2 approaches to writing a project summary. One approach is write it first and use it as a template for the writing of your proposal. So that is, get something down for a project summary that, I love John's analogy. It's one that I tend to use a lot because it's so great. This is like a movie trailer, right? It's giving just enough to excite you about the work to come, but not so much that you're like, man, I don't even need to see that movie because I already know exactly what's going to happen in it. Right? So it's just enough to excite and describe, but not enough to give away the entirety of the proposal. So in that way, like writing it first. So I'm going to just write up here, your choices. Your choice. 1, write it first. as foreplay for your entire proposal, right? And then you look at it and you say, now from this one sentence that I had devoted to, some of these areas, I'm going to then expand and expound upon that as I write the proposal. Two, and very common, is to write it last. So write it first, use it as a template. Write it last, and then in which case you're summarizing what is the larger proposal in a condensed way. So this is like condensation. Right? Oops, I left an N out there. I am getting better with my first grade writing with this tablet. Yeah, they're not easy. Oh my. So this is totally up to you. And the reason that I actually wrote this down and really brought your attention to it is that if you would like to write it first and you want to send it to me by sometime on Thursday, I will try to turn it around and send it back to you by sometime on Friday. And then that way you will have a template going into your proposal writing. If that's of any use to you, if you feel that's the way you would go about it. Who has an opinion already, how you would want to approach it? Use it as a template or just come back later and condense and do it last? Yeah. How come it can feel like that?
Just because I feel like you're more clear
Mm-hmm.
summary.
Mm-hmm. Yeah. Because
If I was more sure of my timeline and plan, then I think
right.
exciting.
More exciting. Exactly. You could focus on the important parts of it instead of, yeah, right. Got it. Yeah, makes sense. Do it last.
That way I wouldn't be asking you to put way too much of everything
Into
that.
yeah.
And then
Right. Instead, you could say, how could I state these 10 sentences of methods in one sentence? How could I condense it, and really just succinctly state it? I think that's a great point, and I think I'm pretty sure that I don't know, I don't want to make a blanket statement, but I do think that a lot of people write it last. I think that's a common thing. I don't mind writing it first, actually, because I sometimes like to have a template to build on, and I'm sort of a little bit more tendent towards wanting to write the interesting, significant stuff first, and then wanting to, jam out the details. So I am totally happy with you guys doing either. It's totally fine however you want to do it. I can tell you something, and that is that, let me just start this. This is reviewed first. So for a reviewer, this is the absolute first thing they're going to see. It's the thing that is going to either capture their attention and give them a clear idea of the proposal to come, or not fail to capture their attention, be dry and uninteresting, or be undescriptive of that which you are planning to accomplish. Okay. Yeah, Jenny.
I know this is different than an abstract all the time, but a lot of abstracts are just, they pull sentences from the body.
Yeah.
And they
Throw it in there.
And so a lot of times you end up reading the abstract and then you're reading the actual paper and you're like,
I read that.
I read that already.
Right.
This should not be that.
Right. Yeah. And I think that what that is, I really do, is sort of a lazy man's work in the abstract. A good abstract really should say it in such a way that it condenses and succinctly states, that which will be much more flowerily described, right, in the proposal itself. So I think that that's maybe a poor example of an abstract. Yeah. Well, I will also say, though, that, when we talk about unifying, right, a big part of that is tell your reader what you're telling them, tell them again, and tell them what you told them. That is true, but it doesn't have to be like these verbatim sentences that you lazily dumped into your abstract. Yeah, no, that's a very good question. So, this is a place to make your work stand out. It's a place to make it memorable. And, it's really an important part of, showing your reader what you plan to do, why it will be influential. So what you plan to do, and why it will be influential. Let's write that down, because that's a good way to summarize this. What you plan to do, and why it will be influential. Okay. Any other thoughts, questions, thinking about a project summary? I love the movie trailer analogy, foreplay to the actual proposal, right? All of those are good ways to think about this document. Okay, so let's do some practice on this. And so I think that we'll be looking at your, if you have your books, we'll grab those out. And we're looking at page 66.
Does this favor certain types of research? I just wonder, so it may be really important to study the mating behaviors of the Asian mole rat, for example. That may be something that's really important.
Mm-hmm.
But I don't know if you could write something as exciting and persuasive
Right.
about that. Or at least trendy, particularly.
Yeah.
So,
Well, and that question does come back, Josh, I think, to that video that we watched last time, right, with the yeast, and it comes back again to the what I hope will be a theme, and I'm glad you keep bringing it up, because I really want it to be a theme through the whole class. Is where do we differentiate between basic science and studying something for the sake of studying it, like the beauty of uncovering what's going on in that phenomena, versus something like we're doing, where like clearly like the impact that it can have on the community is the central focus, and I think both have value. But I really do believe that if you are thoughtful about your research, that one could write a very persuasive significance for the mating behaviors of the Asian mole rat, because I think it's very important to thoughtfully situate your study, and truly, like, if you can't come up with a reason that you're trying to study that, do you feel that is something that, if there is absolutely no way that you can situate that into something significant, I'm not saying, okay, yeah, I'm not saying that it doesn't have, I'm so far away from saying that it doesn't have significance. I think everything does. But I think as the writer and the researcher, you need to think about what it is. Yeah, yeah.
Proposing for funding from the NSF or something that's
Mm-hmm.
so broad, not defined on broad.
Sure.
So just being significant in some niche doesn't necessarily mean that it's always right.
And
how do you define?
how about?
I'm sorry. Go ahead.
No, please.
How do you define something like exciting and persuasive from the perspective of them? So like
Right.
exciting and persuasive
Right.
to me might be
Right.
How do you know
Right.
what your audience would, because they might be trendy. There's
Yeah.
huge research on
Yep.
for
Yep.
example, like Nature, having articles that tend to be very trendy during the time and not
Yeah.
really the most influential, but the more trendy they
Sure.
tend to get in.
Sure.
And so I don't know if I guess to me words like that are very ambiguous.
Sure.
Because I find things that are exciting and
Yeah.
everybody else just stares off into space over.
Yeah.
So I don't know how like my scientific interest, I would define important, or how I would define
Sure.
exciting.
Sure. And the number one thing that I'm going to say, and I don't know if this will be the persuasive answer, but that is that if it's exciting and persuasive to you, and you can properly communicate why you get jazzed about it, I believe it will be exciting and persuasive to any reader. Does that make sense? Like, because to me, now see, this is where I differ a little bit from others, and that is that I believe that if you can do a good job of communicating that, the only place where I think that would fall short is if you did a poor job of communicating why it was so interesting and pivotal. Okay, so take the symbiotic relationship between some exotic, Asian arachnid and some, unique fungus. To me, yes, that has an incredibly broad significance, because the more we understand about the interconnectednesses, right, between all life forms, the more we recognize that imbalances in any tiny micro niche are absolutely pivotally important to us, And so that it's communication, not lack of sometimes trendy, right? I definitely get you there. But I think if you do a good job of communicating why it is important and why you see the import, I think most readers will also see that.
Yeah, well, and then even though you have to write to a general audience, and that's mostly to get
Yeah.
away from just jargon.
Right.
Your proposal is always at least going to be, they're going to attempt to have evaluated by somebody who has expertise in the area.
Yeah.
So if you say this is important to our area because of this and this, and they're going to understand.
They're going to look at that.
Why you're
Absolutely.
looking at what you're looking at.
Yes.
So
Yes.
what you don't want is just this droll list of the things you're going to do in the lab.
Yeah.
And it
And-
matters because we just need to know.
And they do talk a little bit in here about some of the nuances of that. I haven't spent a lot of time on it, but you do have a voice in choosing your reviewers, and if you do want someone within, you don't have total voice, right? But that you do have a little bit of input there, and if you are, it's important to you that someone in your field is at least part of the review panel. There is a little bit of flexibility for.
This is where your title is big.
It's huge, yeah.
They will usually
Use
put you
that
in
to
pools
pool it.
of reviewers based on your title.
Yep.
So if you try to be too kitschy and too cute and don't have specifics of the kind of stuff that you're doing in your title that
Right.
says,
Right.
I'm a microbiologist and I'm doing microbiology
Yeah. Yes.
in here, and it's
Yes.
not reviewed by somebody with that kind of expertise, you're done.
Right.
Makes sense.
Right, absolutely. And that's why, so much of our conversation yesterday, while I know it can seem overdone when we're talking about the title so extensively, but I really believe it's not overdone. Yeah, I really believe it's something we can focus on. So let's take a moment actually to look at page 66, and let's start with just reading through that project summary. Notice that it is the 2-paragraph style that we're choosing to use. They have also chosen to use that style. And if you guys would try to just point out or find in this the things that we've talked about. So let's kick things off with the first two sentences. What do they do with just two sentences there with regards to the areas that we said should be included? What's the role of sentence 1 and sentence 2?
Well, it states the context, the first one.
Good.
And a little bit of the importance as well.
Mm-hmm. I agree, yeah.
That's a little stuck.
Mm-hmm.
To that gap.
Good, Jenny. So sentence 1, I would agree with Laura. Winter desiccation is recognized as an important stress factor in coniferous forests, and it may limit conifer distribution. So this obviously is establishing context somewhat, but importance, I think. So this really is giving that broad funnel of like, here is why we should study this, but then the whole is very much indicated. Most research to date has focused on desiccation at alpine tree line, whereas little attention has been given to its role in establishing the upper elevational limit of low elevation conifers. So they're saying here's what's been done and here's what hasn't been done, and that's establishing the need for research or the hole in research. So those two sentences are really clear for us to see what they've done. What about sentence 3?
The hypothesis.
Yeah, exactly. Like in a nutshell, isn't it? Yeah. Mm-hmm. Mm-hmm. And, you know, we could certainly go back and discuss quality of hypothesis, right? But that's at least what they're attempting to do in that sentence, is to go ahead and state that. So isn't it amazing how much they're adhering to the kind of guideline here of what to do in each sort of small section of paragraph 1? So because then they really do state the aims as well. And then if you look about midway down, well, it's towards the bottom of the page relatively. Notice the sentence that starts with, our approach will use. Right there begins the statement of their methods. I draw your attention to this in a large part because I think that this is a great example of how the methods are stated, but they're not highly detailed. So they say they're going to use physiological measurements, and they give in parentheses what some of these will be, collected near the upper elevational limit of each species during the winter. And they're saying in a nutshell what they're going to do, but they're not maybe giving you the absolute nitty-gritty detail, but it's enough of a statement to give you an idea of what their plan is. What am I going to do with this? And then the second paragraph, you'll notice, really does go into the uses, the possible uses of the work, and the broad significance. So the idea is such an approach sets the stage for future studies of limitations by winter desiccation using other species and under conditions imposed by a changing climate. Broader impacts of this work. I mean, they just go outright and state it. Right? There is no beating around the bush there. You know, they're going to make sure that reviewers don't think they didn't state their broad impacts, right? The use of, okay, so this is where sometimes I think it's funny. Through the use of undergraduate field assistance, we will integrate field science into education. So a lot of that makes me laugh a lot. You know, I'm not saying that it's great that NSF is sort of pushing them to incorporate an undergraduate component, and I think that's neat. It's hard to me when I feel like it's forced that becomes the broad impact, but I think we can certainly see it there. That's unarguable.
I guess another thing, so it seems like there's a lot of corn thrown in. I mean,
Yeah.
people trying to fulfill the exact requirements.
Yeah,
right?
yeah.
Essentially, it's like, hey, we will educate this group.
Yep.
And we're going to make this undergrad do this. And he can be
Totally.
So, do you really have to do that?
No.
Or just people write this crap in?
No, I think I mean, here's where I get to just be on my soapbox and say it shouldn't have to be forced. It shouldn't. Like, I mean, it should be, you know, like when you think about what we're doing with this project, with our research, like, and here we're just these undergrads, right? And me, I don't even have a PhD. But when you think about what we're doing here, what NSF wants and what we're doing here, it's all there. We don't have to force it. I don't have to say, ooh, I could get an undergrad. And that would get, oh yeah, funding, So I guess just, and be aware that is me being on my soapbox. Like, I don't think significance should have to be forced if you start from a place of significance. And does that potentially include the mating behaviors of the, subsurface mole rat of Southern Asia? Yeah, it might. Because maybe it's incredibly significant to, one of the native populations of that area, because perhaps maybe it's something upon which their primary food source relies, and yeah. So I think it's about it's about the place from which we start inquiry. And maybe, if I can say anything to convince you that the place from which we start inquiry is always the most important thing, yeah. Okay, let's look at, see where we wanted to go from here. 68 and 69. And I want to read this project summary, because I think it's wonderful in that it is very, very much in our field, right? We, researchers in our department study C. elegans, and when you're looking at neural network model for chemotaxis in C. elegans, so you think, oh, well, okay, let's check this out. Go ahead and read through that one. Yeah, especially, the latter portion is a little hard to swallow. Let's stay away from that one, because we all know what we're laughing about. What do they do well, though? What are some things that in the earlier portions they do well?
I like the first segment a lot.
I do too, yeah.
I think it's very specific, but it's one of the things they did interestingly I'm not used to is this is the lay public, I think.
Yeah.
I don't know anything about sailing. It's really,
Yeah.
but I can read that and understand.
And feel like you get it. Yeah, I agree with you. I thought that they did a good job of defining their organisms, their model system, their objectives, their aims, their so the first sentence is kind of a broad objective. So they're letting us know what they're going to do. And I think that's well done. Honestly, I think the only thing that, besides picking on, obviously the forced significance is hard to swallow, but I think they could do maybe a little bit better on saying what they think their expected results and uses of those would be. So even if we're talking about, just a very narrow-minded view of that, I think you could still state, here are our expected results and the further uses that we see for those. So so thank you for taking time to...
I noticed that they talk about being sponsored.
Yeah. So they're actually saying that what's the reason for that? Like why? Read us that sentence, Jenny.
Specified versions of the models developed in NSF-sponsored research are a paramount unit of PI's course in computational neuroscience.
Mm-hmm.
What I think is they're trying to say that if we get funded, then this will look good for you guys.
But also, yes, okay, but also they're saying that this work is being used in the classroom in a sustainable model. And that's primarily what they're trying to communicate. And that is significant, because what they're showing with that is that the university is funding and supporting a sustainable course that is based upon this work. And so that has broad and lasting impacts on students. And if funding is provided, it will help to support those broad and lasting impacts. Yeah. Good job in pulling that out, because sometimes it's hard to know, why are they saying that? I mean, what does it mean to them or to us?
So I've noticed a lot of the upper division PIs, when they teach,
Yeah.
often teach the class
Yeah.
very close to their
Yes.
research. So is that a reason why? I imagine as a researcher you get
Mhm.
hyper good at what you do
Yes.
and everything else. So teaching that just makes sense. But
Yeah. Yep.
Is that another reason why that happens?
Yeah.
Because then you could, it seems like in grant writing you can say my research
Yeah,
is-
for NSF-funded faculty, it is. And so we don't have a lot of NSF-funded faculty in our department, but Mark Gomelski is, and very interested. And I have been, I'll be honest, it's been very exciting to watch him take a greater and greater interest in undergraduate education. And if NSF is helping to push that, while maybe it's forced a little bit, it's been really awesome to see that increased, because the quality of the teaching goes up. When you're able to couple it with something you're passionate about, I think the quality stays high. So, that can be done both good in both good and bad ways. But yeah, it's a great question.
Yeah, it was good.
Yeah.
Mark's class was very similar to that.
Yep.
Research was excellent.
But it's excellent. It's become excellent. Yeah. And I think it's a great question, and I think, that it is that the effort that's being made there is important to you as a student. Yeah. Very good questions. You guys are really getting at some of the finer nuances of all of this, and it's so very important to helping you as you go into graduate work, I hope. All right, really quickly, I just want to talk briefly about the timeline, and I know that I kind of stayed out there. The timeline is really just a proposed plan for your project. And essentially there are a number of ways that you can do this, but there's a lot that you need to consider in the timeline. You need to consider, for example, when talking with Leanne and how she said, hey, when are you going to need to do these C to N ratios? We need to find out when it's convenient for her. And so in our timeline, we would want to base it partly on when would be a convenient time to take those samples to her.
She said milligrams there.
Yeah.
Keep at least 5 grams.
5 grams, I would say, yeah.
So milligrams are what go in the analyzer after she grinds it down and pulls out what she needs.
Yeah. So
10 times at least.
Taking her into account, her schedule, right, and scheduling with her as a part of your timeline. Also, thinking about sampling. Now, for downtown clinic, you guys, you're going to have the probiotic, but you might want to go to the wastewater treatment plant, right, and look at potentially, collecting samples there. How are you going to schedule in the time for that sampling? Where will that fit? So our timelines, when you submit your individual proposals, will be very rough. But as we work through them next week as a group, I hope we can turn them into something much more solid. So, things like equipment, scheduling, sampling, culture growth, what are all of the things that we need to take into account? And one of the things that we're going to notice, even starting this week and certainly next week, is that some of our bacteria will take 48 hours to grow. So that might mean that you can't put in a culture on Monday and hope to follow up on it on Tuesday. You might have to schedule in such a way that you're doing things like 48 hours later. So you guys might have a plan where, say for acres, you might have a plan where you go and do your coring out at acres on Fridays, and then maybe you come in and quickly plate them out so that you're ready to go on Monday. Or, I don't know. It's going to be a matter of thinking through what will be the best approach for allowing your samples to be grown and ready to process when you really want to be able to do that. The thing that we often say about a timeline is that science and reality meet in a timeline because, and John will attest to, I'm the dreamer. I want to be able to take on the world. I want to be able to show the most significant things. But when you really start to see that meshing with timeline and the 6-week period that we have to do research, give or take a little bit of time on each side, we don't have a lot of time. So we sometimes have to think about reality meeting, how we're going to hit the pavement there and how that's going to maybe limit us to a certain extent. But this is one really great way to make sure that your budgeting is realistic. And I know I'm not asking you guys to submit a budget, but that is often a central part of a grant. And when we reviewed those RFPs, how we said, budget seems to be really important to them, how you're spending the money, of course. And this may help you to write a more realistic budget. And it shows, whether or not it's a feasible study, like really helps you to get onto whether that feasibility is there.
Or for you guys, when you're talking about sampling and culturing and things like that, if you were to say, we're planning on every day of the week to plate 300 plates, you'll see that that's not feasible, because it means that every day of the week you're also happy to do it. And then the next day you're happy to count 300 plates, plate 300 plates, and make 300 plates. And then at some point in time you've got to do your decon, you've got to do your additions. And so, hopefully in putting together your timeline and ultimately your project methods, that you can see that this is realistically done.
Yeah.
So to say there's going to be 10 to the 9, which means I'm going to need 9 plates for each of these different things. How are we going to do that?
Mm-hmm. Yeah.
And tubes that have to be sterile, and PBS, and you got to pipe yourself. There's a lot of reality that's going to be hit in terms of what you can do when you start sorting this out.
Mm-hmm. Yeah, and I think, maybe one of the most difficult things is to take what's realistic and operate within the confines of that so that you're not going crazy. And I think if you stay in a realistic sort of framework, it does enable you to not so balance, right? Not lose touch with significance and what you're really trying to do, while at the same time as staying pragmatic. So it is a fine line, but I think the best research is done when you do limit yourself so that you're able to accomplish what you want to accomplish while at the same time as keeping focused on what it is that is really important, rather than getting sidetracked with, the 300 plates that you think might be helpful, maybe, to try to get it, wheedled down to something that will give you some workable results, even if you're not taking on the world. Questions at all on timeline? Let me ask you a question. What is even just one thing, besides I mentioned scheduling the seed and ratios, what is one thing for your group that you think would have to go on your timeline that might be a limiting factor?
If we opted the sequence,
Yeah.
that sets a limit on how long it's going to take her to process,
Right.
as an example.
Right. So we would want to ask and just see, hey, when do we need to get those samples to you? What would be reasonable, and then back plan from that as to when we maybe need to do those, DNA preps or whatever it is that we would need to have the DNA pure and isolated. Yeah. Good. Excellent. What about you guys?
The big one that you mentioned would
you
These
know.
are going to take different amounts of time to culture.
Right. And especially you guys are looking a little bit at trying to look at some of the fungal population, you know, maybe thinking about how long, you know, that will take to culture. And then, you know, and depending, you know, finding the optimum culturing conditions for that. So time-wise preparing that.
Also, if you have to isolate anything naturally, so because you guys are working on getting seeded right now. And so the other option is always, and this is typically what we do in our lab because we don't have money and time, is that you just isolate it from nature. But that adds 2 or 3 weeks on the front end verifying that you have what you think you have.
Right.
And that significantly changes the time.
Mm-hmm.
You say, I've got 6 weeks and I'm going to spend at best 2 weeks on a short end identifying the kind of thing that we want to test against. So on that, Rachel sent out that study that you guys saw that had salmonella in it as well. And so if in addition to C. diff, or if we can get it relatively easily, and it's like $300 to buy, so like that's manageable, but it cuts a lot into what we want to
C. diff, Capstone?
Yeah, from ATCC. So if you guys want to do other typical foodborne pathogens like salmonella or E. coli or some of those kind of things, we have a lot of them that should go on. So if you wanted to look at the probiotic and its effect on the general like foodborne illness kind of causers, that might be an option to go with. I mean, I think, you know, looking at this, especially with the population we have, C. diff is preferable. But in terms of rubber hitting the road, and if you say, like, legitimately we just can't have it in-house, we're going to start at something where you're going to have to isolate it, you're going to do something to do besides waiting and growing. It's being in those first 3 weeks, and so, you know, you might be doing some of the plate-based inhibition assays and things like that with your probiotic during that time while you're doing your C. diff. So do know that is an option, but I have a ton of that kind of stuff running around as long as you're not averse to working with pathogens. Which does change a little bit about how you have to
How to handle
manage
them.
yourself in the lab and like everything does have to go back and stuff like that.
We have been working on seeing if Jerry has C. diff and Dr. G.
No one else on campus, I think.
Yeah.
Could we call around maybe a little bit? Because I could even do that. I know for the right reasons, a lot of the colleges, big professors will swap too.
Right. No,
So
the
I
researchers
just-
will do it. So, but if they, well, it's a BO1, so it might not be a shipping issue. Yeah, I mean, like we can.
Right.
We can look.
Okay.
But
Wouldn't
it's
be
always,
too much
it's
of
always
a shipping
easier
issue.
if you don't have to ship it.
Sure.
Yeah.
It
Even
becomes
if there's someplace
problematic.
close.
Mhm.
Yeah, and before Collins,
Yeah, and that used to be a pretty easy thing. And yeah, being a class 1, I think you'd be.
Yeah, well, so it's.
What's the story there?
It depends on the institution.
Okay.
Typically, as to how they classify it.
Okay.
Where it can cause disease.
Yeah.
Technically, it should be a BL2.
Yeah.
ATCC has it listed as a BL1.
Okay.
Which is easy, but anything that's a BL2 or above, you have to have a shipping certificate that says that you can ship it. You have to have a receiving certificate that says you can get it, and technically it has to be CDC approved.
Right.
That's long.
It takes a long time.
You know, so, but there's, so anybody receiving it here has to be up to date on their USDOT shipping and receiving certificate,
Right.
and whoever's shipping it has to be.
Right.
And those classes aren't like regularly easy to find.
Right.
So, I think that I'm close. I don't know that I'm up to date though. If it's 3 years, I am. If it's 2 years, I'm not.
I know I'm not up to date.
So there are logistical
Yeah.
issues in shipping. Now, if you're driving it, you can physically, if you are in your vehicle, drive it across state lines.
Right.
But you can't have somebody else transport it for you across state lines.
Right.
I mean,
So
there's...
whoever requests has to get.
Yeah, so like
Yeah.
if we were gonna ask for it, like Rachel or I would have to drive to Boulder to pick it up, to bring it back. Because it's then going to go in our lab in our user. So it's pretty highly regulated. Now, I mean, again, like I said, if we send Josh and he brings it up here, it happens all the time, because what's the likelihood that Josh is going to get in a car wreck and
Yeah.
You never know, but you know, if you're driving and in the backseat of your car you've got a cooler that has a biohazard sticker on it, and Josh is driving like an ass and he gets pulled over, then you're in trouble.
That doesn't look very good.
You want to drop the bag of weed at that point so that they have the red herring. It's easier to deal with, especially in Colorado.